Okay, everyone, maybe we should rejoin. Let's see who is present. Could you please switch on your videos now, if possible, so everyone can see you? I think the audience can see the person who is speaking. So as far as I know, and otherwise, please correct me, Franzi, it's always the person who speaks who can be seen and not the whole group. So we have received a number of questions from the audience that Dina and I will ask you. We will not be able to address all the questions, but we'll select a few. We'll want to give each of you an opportunity to address some of them. Please keep it short if possible. And do feel free, however, we have 30 minutes left to embark also a little bit on more general issues about Huntington's disease treatments. I think the topic of this session is also of this panel discussion, what are the lessons learned in the drug development process that you have undertaken in your projects, that you are undertaking? What are the issues that you worry about? And in the context of answering your questions, feel free to address these issues as well. So Dina, do you want to start with the first question?
Yes, I just want to make a general statement to say thank you for the speakers, all very interesting. Clinical trials is in the forefront of the HD community, and I think we can consider ourselves actually very lucky that there's so much going on. It may seem like a slow process, but there is a lot, and we should be grateful for all of this. So I'm going to start with the first, and we're going to, as Ralph said, we got a lot of questions, which we're not going to be able to get through. So we're going to first go in order of presenters. And first question is for Scott. Should I say who the question is from, or is that not important?
Whatever, the question is fine.
Yeah. So the question is, glad to hear that Roche intends to share the generation HD1 data with the broader HD community. When approximately will this big data be available? Which data will be shared? And is there already a procedure in place for requesting data, or will this be determined later?
Yeah, sure. So in terms of data sharing, I think towards the end of the year, we're looking at getting together data interpretation on the continued safety outcomes of the participants of the ongoing studies. And then beyond that, into early next, I would say at some point, the whole integrated view in the company position on where we're going with tominersen. That's the overall timeframe. It's very difficult for me, though, to commit to something more specific than that, given all of the stakeholders we have to manage, internal and external, and the fact that the data is not ready. We're literally still getting in fluid biomarker data, for example, from our external vendors, so it's just hard to say. But that's the general timeframe. And then the second question in terms of sharing like data with the community more directly. Generally, once trials are completed and we have a clinical study report, there's a mechanism of investigators to request data, for that kind of investigator-initiated analysis. And I'm sure that's something that we have a very open mind to and have done repeatedly on different programs at Roche over time. But that generally doesn't occur until trials are done. And even then, it might be when the programs are going out of phase 3 into phase 4 or are alternatively closing. That's a time typically when that kind of information exchange becomes more possible. So that's the general answer to that question.
Thank you, Scott.
Thank you. So I will proceed with a question for Maurice. Maurice, there is a question concerning the BSI analysis that you presented as percentage change from baseline. So it's noted that at baseline, no compound was present in the system, obviously. So the question is, how do you address the possibility that apparent attenuation of caudate volume loss represents swelling of brain tissue induced by the compound? So does the apparent attenuation of atrophy rate persist if you take the first MRI in the presence of the compound as baseline, and then you look at the trajectories? Have you looked at that?
Yeah, so I don't think that's a concern. We had 3 time points. We had baseline, 6 months, and 18 months. And actually, most of the spread between drug and placebo was between 6 and 18 months. And so during that time span, both there was drug present at the 6-month time point in presumably steady state concentrations.
Mm-hmm. Okay. Thank you very much. So Dina, do you have a question?
Yes, a question for VZ. It's a question that came up, but it's actually also a question on my part, and that is, how frequent is the SNP for wave 3 and in different regions, and were your hopes aligned with reality in the recruitment of SELECT-HD?
So what has been reported in the literature are in the range of 25 to 40%. So we are refining that analysis actually, because it may be different in different regions. So we are opening up regions where we weren't present before, such as Italy and Spain, that seem to have a higher prevalence. But we are going with what the literature says at the moment. And at the moment, we have found basically around 33%, I believe. I don't have the exact calculation. I don't want to give you a number, but the prevalence is different, again, in different regions.
Okay, thank you for addressing that. Another question now, maybe for David. So I'll go around first one time. David, there is a question concerning the possible evidence that you talked about for the virus vector to have anterograde or retrograde distribution after injection. So can you address and elaborate a little bit more on that? How much do you know about it? How reliable is the data, maybe from the guinea pig models and the data you did in you got from large animal models for anterograde and retrograde transport of AMT in the intrastriatal injection, after the intrastriatal injections into other brain areas?
So as a neurosurgeon and clinician, I'll keep this short. I think there's 2 sources of the evidence. One is cell culture models where we can actually look at it. The second is, as you mentioned, the large animal models where we can see where an intrastriatal injection ultimately ends up distributing. So we can look at more at the mechanistic side of how it's distributing in the cell culture models, and we can look at more of how much it distributes into what areas of the brain and along what potential pathways in the large animals. And we certainly, with 56 mini pigs and non-human primates, have a lot of animals followed out to a long period of time now to look at what other areas of cortex, brainstem, et cetera, all show expression, and therefore must have gotten the vector from someplace because we put it in the striatum.
May I follow up on this briefly? So how long have you followed up the mini pigs so far? What's the longest?
The mini pigs are, I think the longest is out to 3 years now.
And how many would that be? Can you elaborate on that?
I can't tell you the exact number.
Mm-hmm. Okay, thank you. Dina, you have the next question.
So the next question is for Irina, and the question is, will you take the MSH3 SNP status in deciding which patients to include in your planned study? Some MSH3 SNP variants are predicted to lower MSH3 protein levels than other SNPs.
Yes, absolutely. So of course, this is something that is on our mind. We actually have the SNP data for SHIELD-HD already. So the SNP distribution is actually, again, as expected in the literature. So yes, we have some SNPs in MSH3. Given that the published data indicate that the MSH3 expression, even with the SNPs, is still only about 15 to maybe 20-25% lower than in the wild type, we think that treating patients and inducing at least a 50% sustained knockdown in the brain is still going to have a potential to have a therapeutic benefit. So we will not necessarily exclude, or we will not exclude patients based on the SNP profile, but we will collect the SNP profiles, and we will be looking at patients and if this is any covariate that is relevant to include when we do the analysis. And we will be doing this also in SHIELD HD, where we will look at the disease course in the context of the different SNP profiles.
Thank you.
Thanks, Irina. So, Michael, we don't have any specific questions for you as of yet. So it seems the presentation was very clear, and obviously we've been doing the development for a little longer. But I have a question to you. Do you think that the overall, like the duration of the study is going to be sufficient for us to address potential disease-modifying effects in the future? How did you arrive at doing it at the current duration that you were looking at for 1.5 years?
So I don't want to use the word disease modification because we're seeing an impact on TFC in prior studies, but we don't understand precisely, we don't have long-term data in a controlled way to talk about disease modification. And furthermore, if we were looking for that in this trial, the trial design would be quite different. We're looking for maintenance of functional capacity, and patients will have to follow long-term in an effort to see that. Now, the reason we chose 65 weeks is that certainly in early HD, there is good evidence from so many studies that you will get a decrease between 0.8 and certainly around 0.8 if you combine stage 1 and stage 2 for 1 year. So we've gone longer than that in an effort to further allow placebo to go down. And furthermore, we're very well powered. So this study, and this is powered, we initially powered for a 20% dropout. We've had nothing like that. We've had under 5% and less. So we're very well powered to see something in TFC with this based on prior data and at 65 weeks. And certainly we've also seen early preliminary exploratory evidence that NFL may also be an important biomarker in the study. So we've been able to take all the primary and secondary endpoints have been shown in some studies before to have the potential for significant results. The regulators have accepted this duration. This was a big discussion with the regulators. And so we wanted to essentially give ourselves the power to see the effect based on power of real clinical data. But if this has some impact on TFC, then we also are going to continue to follow patients in open label for an extensive period of time to really see the duration and durability of the effect if we're able to show it.
Yeah, it's obviously a challenge, I think, anyway, for all of us, right, to find the right time for study durations. I think this has been discussed so much in the community. We've made maybe some, let me ask you this way. Right now, maybe that's not the only factor. Let's see how much time we have left to discuss specific issues afterwards. For each of you, what would you have different? What would you have done different in your program if you could now, looking back at the trials that you have done, looking back at the results you have obtained? What would you have done different? What do you think lacks right now that you could have gained? What would you do different next time? Maybe we'll start with Scott.
Yeah, Ralph, thanks for the question. I think, one main thing I think that we've learned is, and it's really a fundamental lesson of drug development and pharmacology, is I think we were so enamored with our phase 1 results and the dose-dependent lowering of CSF mutant huntingtin that we naively assumed that taking the top dose forward into chronic studies would be fine, and that we would run into no issues of tolerability or problems with that. We also kind of similarly thought in terms of the monthly frequency, and I don't think we appreciated just how potent the drug was. I think if I had to do it over again, or we had to do it over again, we would have dose ranged and frequency ranged much earlier in the program. Again, it's a fundamental lesson of drug development, but I think there's been such excitement, it's not only in this space, but in monogenic diseases generally. Like there's a lot of talk in papers, white papers out there on phase skipping, skipping, phase 2 and going straight from phase 1 to phase 3. I think that though, in our experience, has sort of not been a good choice. So that's I would say the main one, if I had to answer your question.
Thank you. I think it's an important lesson that we may have to learn, right? We were all very enthusiastic, as you say, about the biomarker data that we saw, but maybe it is, after all, something to consider that in phase 2, we want to potentially see a dose-dependent clinical effect as well, at least an exploratory clinical effect before we embark in huge clinical trials. Good point. So maybe Maurice, what's your
or Rob, just let me respond back to that, or at least a threshold at which you start to see clinical efficacy. I think dose-dependent clinical efficacy may be a high bar.
That's why maybe some.
Yeah, but I'm with you. Yeah.
Let's say quantitative or whatever it is, but see some clinical correlation to the biomarker data, the proof of concept biomarker engagement target that we might have seen. So it would be nice to see something along those lines, right?
Yes.
Possibly. Yeah. Okay, Maurice, what's your lesson learned from the Signal trial? I mean, you've done multiple arms. It was a very complex design of this trial with different analyses that had to be done. So is there anything that you would like to share with us openly that we can learn for our next design?
Right. So as I alluded to in my remarks, a very important issue for us is what's the right stage of disease for the mechanism of action of your drug. And we did not have enough information about that when we started this study. When I think about it now, you ask yourself, what happens during early disease progression? And two broad models are, for example, there's a chain of events with multiple discrete steps, each one leading to the next. A, B, C, D, E, for example. And maybe your drug is relevant in stage D, and you need to time that accordingly. But there's another model, which I think is probably closer to reality, and that is there's repetitive oscillation of two counterbalancing responses that, as a result of disease-related disturbances, fail to reach a stable equilibrium. And this is really closer to what we know about how, for example, the immune system operates, where you have an aggressive response and there's a counter reaction, an inhibitory response to the aggressive response, and they come to some kind of a balance, and that may not be adequate for responding to the disease symptoms. And so you have to intervene at that point. And this is what's going on in cancer research with checkpoint inhibitors and so forth. And I think that it's not unlikely that there are similar interactions in the central nervous system.
So we could learn from those fields as well. I think it was interesting, by the way, over the last few years that we have oftentimes had the opportunity to listen to speakers in our conferences at EHDN, at CHDI, and HSG that came from other fields and brought in some experience that we could take. And yeah, thank you, Maurice. So how about Visha? What's your lesson learned from the previous studies? Now, there were 2 that unfortunately had to be stopped so far. And what would you do different next time? I mean, there was a challenge, I think, also here, and that may be an issue that we can also address briefly, everybody's point of view about animal models, particularly because with your SNP1 and SNP2, you didn't really have animal models.
Absolutely, the big gap that we had. We didn't have a robust preclinical models, not only to measure the pharmacodynamic effect or target engagement, but even PK distribution of the drug, which seems to be quite important now in light of the Scott's results, to determine frequency and dosing of our drugs. So we hope at least we have covered that gap. But another one that I think it's common across the board is the reliability of biomarker measurements and the ability to measure them on an ongoing basis so that one can adapt trials even when you are in the clinic. So we really need to put a concerted effort into developing biomarkers assessment that are reliable across the board and are comparable across across drugs and assets.
That would be helpful. Yeah, thank you for sharing those insights.
So just to comment, though, even having robust preclinical data, which in the Roche trial they have it, you still did not have the outcomes that that we wanted to see. So that's.
We always have to get proof of concept in the clinic in the end, right? In humans.
My view is a lot of times that animal models are great and they have to be done, but a lot of times they don't step forward into the human diseases and how the human body responds.
We've had that challenge quite often, haven't we? David, what's your feedback to us, although you haven't been in the Huntington field for too long yet, not too many clinical trials, but what's the challenge that you have experienced, or what's your, I mean, obviously there were some challenges around the selection of the patients also in the early studies with the CAG and the brain volume issues. You're tapping into a total new territory for Huntington's disease clinical trials. I think the community needs to learn from that, but what's your feedback to us?
I mean, I would say that's one huge issue that we had in terms of what really is the predictiveness of any of the natural history data when you go look at subpopulations like by MRI initial volume, right? It's the first trial that's ever had a volume threshold for getting in. It creates different expectations for what you get out on the other side and what the population is you need to recruit. I think the other part of it that's really interesting is the surgical side. I mean, you really coming into a first human dose as a neurosurgeon versus a medical neurologist, and that level of detail that you need to get to operationally, not only in the OR and modifying the procedure with every procedure done and lesson learned, but also on the back end with IMP preparation and what is it actually going to take to get a pharmacy to be able to do this and do it successfully without any issues. So, I mean, we've constantly been tweaking the manual for both administration and preparation, and debriefing extensively after each case. So it's a real learning experience that you can take into other programs. And I think one of the big gaps there is most of the programs in Parkinson's, the gene therapy, cell therapies, et cetera, have ultimately failed, and little detail is out there. The FDA advisory committee a week ago was talking about MRI findings with basically no neuroradiologists on the call, speaking to what are the established findings you'd expect in imaging post-op, and most of the animal models are, not so relevant. But that getting a lot of observations is good, but knowing how to interpret a lot of the data that you generate in the early post-operative period is also, apart from clinical and safety considerations, which are pretty easy for DSMB to look at, a lot of these other biomarkers around surgery are very poorly characterized. And even though we've been doing gene therapies for more than a decade, these things are very poorly characterized.
So a lot of learning to be done here, right? In the next.
And the importance of communicating that data afterwards, even if the program is unsuccessful, people need to be communicating. What does an MRI look like for a year, 2 years, 3 years after gene therapy? What does NFL do? What do all these things change? We have very minimal data from Nurturin and some other very early programs.
Yes, I think you already had some learning that we saw in your trials, obviously, and it will be great to share that with everyone, of course. Okay, we don't have too much time left, so I want to give everyone the chance. Irina, what's your message to us?
Well, I mean, obviously we have not started our intervention study. So I think for Shield HD, we were just very fortunate that everything so far seemed to be going quite well. But I would say what others have also said, I think for us this is very important, is to really have, for instance, a solid PK/PD model, really understand the biodistribution and the relationship between biodistribution and knockdown. So to really be able to have a strong hypothesis going in on what dosing will be needed. And then with the clinical biomarker and the clinical PK data, check that our hypothesis was right, and if not, change. But I mean, one lesson I would say is that we have relied on intrathecal based on, other studies. And it is only when we really took a hard look, and I was for sure not the one who took this easily. I mean, I know that I was like, I really wasn't sure that we could abandon IT. But I think all the data we have seen since then internally, but also published, I just literally do not believe that intrathecal delivery reaches the deep brain of an unconjugated ASO. And once I have adopted this notion and accepted this, then the door was really open to explore different routes of administration. And I strongly think that they are much more feasible than I had initially thought. So it's a patient population that deserves the best shot we can give them. I mean, every patient population deserves that. And I want to be absolutely sure that we can deliver to where we need to go. And of course, do it safely, but this is the clinical study that we should.
So we are taking different steps here, and that is great. I mean, what a variety of different approaches we have with you guys represented here in this single panel, right? It's amazing. And it's, as
Ralph, if I could just respond to a couple of those comments. I applaud your pursuit of ICV. I think it's great that you're doing that modality, but I just wanted to clear up a couple of statements you made. On the PK/PD front, we've had a very robust model which has predicted our CSF lowering well. But the message I was trying to give earlier, and perhaps I wasn't clear, was we just simply didn't anticipate the longevity of the ASO effect, but we know to a certainty, at least in our large animal studies, that we do reach regions like caudate. But and I just wanted to say also on the tolerability side, you know, we've now done 10,000 or more intrathecal procedures, and we have you know, a tolerability rate. We have mild side effects in about 10% of participants, like, and it is back pain, as you mentioned. We have no evidence of scarring. We've looked at that with enhanced gadolinium imaging systematically as well in a cohort, you know, along the way. So I do think that intrathecal is tolerable in a very large scale chronic setting, you know, 10,000 procedures in our program, it's going well. But I still think the fundamental question of, is that the best modality, you know, relative to like an ICV approach, that is a legitimate question.
I think we will
I just don't want to prejudge it. I think it's premature to speculate, you know, on whether one is necessarily better than the other with a given modality, just to make that statement. Yeah.
Ralph, may I comment?
Yes, please.
Let me just firstly say, what did I learn? A lot from the prior PRIDE study. And perhaps the biggest lesson was extrapolating from the findings in TFC to talk about clinical progression. We didn't really, and we don't have the data, and the design wasn't designed to assess in the right way clinical progression. But let us be clear, the maintenance of functional capacity would be of huge impact to patients. To stay working, to stay managing their finances, to be able to hug their kids and take their grandchildren for a walk. These are important, but it may not be mediated by disease modification. It could also be symptomatic, but that would be still a huge step forward. That's number one. And then number two, I want to make the plea. You know, we're trying to treat Huntington disease, not just for Europe and North America, but for the world. And access is really important. And that's also why I believe oral approaches with a drug that's diffusible, not just pridopatine, but other approaches that allow this drug to be distributed easily. The infrastructure for intrathecal and ICV, really exciting work being done, but the infrastructure there for widespread universal delivery is just not there. And so as we look at this, I would say we need to let multiple flowers bloom. We need to, in the end, they're going to be combination therapies, and we need to be open to different forms of therapy, different combinations, in an effort to give most patients around the world the best chances.
Michael, I think with your long experience in Huntington's disease, that's already a good summary. So maybe, however, Dina, if that's okay with you, I have one proposal. We only have 3 minutes left. So could each of you forget what companies you're working for and what your current project is, and tell us in 20 seconds, what's your big wish for Huntington's disease? What would you like to have available in 1 year? What do you need? What biomarker? What is it? What do you need? What are you dreaming of? If somebody would come along and say, I have $10 billion, I give it to you tomorrow, get this done. What would you like to get done for Huntington's disease? And we will go in reverse order now to make it a bit more stressful for these people that usually were at the end. So Michael, what's your wish? 20 seconds.
I'd like, outside of the drug we're working on, other oral drugs that have the opportunity to be used in combination and could be available globally in an effort to have impact on the maintenance of functional capacity and keep families together. And hopefully in the long term, we show a slowdown of clinical progression.
Great, thank you. Irina, it's your turn, 20 minutes, your wish list.
20 seconds, I think.
Sorry, 20 seconds. But I'm sure you could fill 20 minutes as well.
I mean, of course, I echo what Michael just said, but for us immediately, it would be important to have a regulatory acceptance of using outcomes other than TFC. So clearly going after the pre-manifest patient population. We do know that the disease doesn't start at clinical manifestation. We do want to have a robust outcome that we can measure that reflects that there is a benefit, if there is a benefit, and that the regulators will accept. I think, I know we have the CPAS consortium. I hope that we can get there earlier rather than later.
Thank you, Irina. Clear statement. I agree with you. David, what's your point to make?
So I would just add to that. I mean, I think to me, it's having a full set of imaging, clinical, and biofluid biomarkers that are backed up by natural history and that are all, I think we've all commented on the stability of a lot of the assays, et cetera, that are all very stable on very fixed platforms with best methodologies optimized, right? Like, I think part of the challenge is we're all optimizing as we go with each trial, learning more about the different imaging sequences, the different biomarkers, how to analyze them. Having all of that up front and not having to figure that part out as we go would let us focus on the drug and how to make it work or how to optimize how it works.
Thanks, David. I agree as well. So Visha, your turn.
Yeah, I might not be very original here, but more sensitive clinical outcomes that we can deploy across the board, especially when we think of moving earlier and earlier in the onset of the disease, where the established, validated clinical endpoint may not be suitable, as well as biomarkers that may predict our treatment effect early on and de-risk these large programs that we put in place.
We are hearing this similar messages here from everyone, right? Maurice, what's your desire? 20 seconds max.
So I like Michael, I think that the path of the future is going to include combination therapies. And in order to know which combinations are going to be effective, we need a better understanding of the pathogenic mechanisms of the disease and how each of our drugs affect different aspects of that. And so I feel a deficit in understanding the disease.
Scott.
You know, I just want to say to me, to the community, to patients, families participating in research, keep the faith. I think we've had a couple of false starts over the last year, but just seeing my colleagues here present in this very panel, you know, triplet with its observational study going towards the clinic, Marie's still going, you know, SNP3, Pridopidine getting a fair test. We're still prosecuting our data to the best of our ability. I'm extremely excited for the potential of this field. And, you know, it may take a little bit, we may have to persist, but I just see the evidence from my colleagues here on this very call that grit and persistence is there. And just keep the faith, you know, keep participating, it's worth it. And we're gonna get the answers, I believe. So I just wanted to say that, thanks. Yeah.
So I just want to echo everybody. As an HD family member, I definitely am keeping the faith. It does seem like a long road, but we will get there one day. So if not in my lifetime, but at least in my children's lifetime. So
I think we all admire the patience of the patients. So it is amazing how much you and your families put into this. And I've been myself now involved for 30 years in this field. I'm very optimistic though, when I see what we have been talking about here today, what I can commit to from the clinical side of Huntington's disease physicians. I think together with the families, we are working on changing the diagnostic criteria for Huntington's disease to give an opportunity for people to get treated early. I think we've learned that over the years with all the research that Huntington's disease starts a lot earlier than it's currently declared as manifest disease, and this will open opportunities for treatments early. I think we learned that's important. Thank you all for this wonderful open discussion, for your contributions. And I'm really looking forward to getting more clinical trials reported and discussed in the future EHDN meetings, hopefully live next year. Thank you all.
Can I just make one comment that is there were a lot of questions that were asked, and if you could log in afterwards and maybe answer the questions that to each one of you from the participants, it just wasn't enough time to go through everything.
So
Exactly.
thank you all. Thank you to the panel for that really insightful discussion. It's an indicator of how far things have come over the last few years that we're now having these sorts of discussions. So we now have a short break, back again at 16:45. Just as a reminder, when you return from your break, you'll need to manually click on your chosen parallel session. It won't happen automatically. So please enjoy your break, and we'll see you in the parallel sessions in just over 10 minutes. Thank you.
Bye-bye, everyone. Thank you.
Thank you. Bye-bye.
Thank you. Bye.